Okay, so sometimes you can have, you can change things, and it looks like you've actually may have had recombination occur, but there's really maybe something else going on. So in this, what we're going to talk about is what gene conversion is and how it can occur via mismatch repair. Now, the causes of gene conversion, there's two, potentially. Two ways you can get it. Genetic recombination can cause the different alleles to become identical alleles. This can occur via gene conversion. It's a process whereby one of the alleles is converted to the other. That itself can occur in one of two ways, either via mismatch repair or this gap repair synthesis. And this gap repair was something that we talked about before in that double-stranded break model as a way to get that strand, replace the pieces of the homologous chromatid strand that had been degraded when prior to invasion by that strand into the non-sister chromatid or the homologous chromatid. So here we're just going to focus on DNA mismatch repair. Now, in this case, in this figure, I just want to remind you that only two of the recombining strands are shown. That means those other ones are gone away. So whether it's the holiday model or the double-stranded break model, we've gone through those initial steps to form, so that we've gotten to the point where we've formed our holiday junction and we've got branch migration occurring. So we're at the third or the fourth step, depending on which model you're talking about. We start out with two homologous chromatids, and they differ by a single position within this region. But these differences are enough that they cause different expression of this protein, so they're considered different alleles of the protein, or different alleles of the gene. So now we're going to go down. We've got the formation or the creation of the heteroduplexes as indicated here within this box. There's a couple of ways that we can resolve this mismatch that we have on both of these homologous chromatids. On the top, we've got a GT pair, or a G and a T that's trying to pair, and then on the bottom we've got an A and a C, and they're trying to pair. And we know that these typically don't bind or don't form bonds between them because A only has 2 available hydrogen bonds and C has 3. And up here, G has 2 and T only has 2. So these creates this mismatch or unstable binding. Now, we can repair or change these, fix these mismatches using a DNA repair mechanism. However, what you're going to get are a bunch of different combinations or different ways. There's different ways that you can do it because you're not sure which of these, if this, like for this top strand, whether you need to replace the G with a, or whether you should take the T out and put a C in, or down here in this bottom strand, whether you should be taking the A out and replacing it with a G, or taking the C out and replacing it with a T. Those are all the possible ways that you can fix it. So however way you choose, or whichever way is chosen to repair this mismatch, depends on whether you get gene conversion or not. So remember, we're going to be comparing these strands down here to these homologous chromatids up here. So if we repair, if for this one, this top one right here, if we take the T out and replace it with a C, and then for the bottom strand, if we take the A out and replace it with a G, then we've got gene conversion occurring. Because what we've done is taken the bottom strand that up on here And this original homologous chromatid actually was an AT pair. We've now converted it to, we've now converted it to a GC pair. So we've gone from AT to GC. Now if we look at this one, this right here in this big block, and we compare this to what we had originally, notice that we've still got a GC pair in this top one, and in this bottom fragment we've still got A and T. So we haven't converted anything. This top strand is the result of removing the T and replacing it with a C. The bottom is a result of removing the C and replacing it with a T. So we kind of repaired the mismatch and brought it back to the original. Again, we can have no conversion, no gene conversion, if we take that top G here and replace it with an A, so we have AT pair there. And if we take the bottom A here, and replace it with a G on this strand, that means that we still have the two alleles present. We've just switched the location of them essentially, but it's not considered gene conversion because they match the original homologous chromatids that we have up here at the top. The final way that we could repair it is if we take out the C on this bottom one and replace it with a T, so that we have an AT pair down here, and we take out the G on the top strand and replace it with an A, so that we have an AT pair up here. This is an example of a gene conversion, because you notice, as just like with this one, now we've removed one of the potential alleles that was found in the original homologous chromatid. So now we've basically reduced the genetic diversity in this situation. So gene conversion can have a profound effect on diversity, on genetic diversity, and it's mostly because there's so many ways, or there's so many possible ways that you can repair or fix these mismatches that occurred in this heteroduplex region. And I've just drawn these as the mismatches that are actually having to be replaced in either of these homologous chromatids.